Hello students, welcome back to the biology class. So in the last class we continued the 19th chapter, that is the excretory products and their elimination, and we learned about the blood supply to the kidneys. We learned that it is the dorsal aorta which produces a branch which finally leads into a renal artery, and this renal artery enters into the kidney, and later it branches again into numerous branches, and each of the branch will finally enter into the nephron. We learned in the last class that that branch is called as efferent arteriole, which carries the blood for filtration, and the efferent arteriole later branch into around 50 to 100 tuft of or a collection or a bundle of capillaries, which are specialized capillaries in the kidneys, and they are called as the glomerulus in the Bowman capsule. And from there the blood, once filtration is over, the blood passes out into a narrower tube in the cortical nephrons, and that narrower tube is because to increase the BP, and those tubes are called as the efferent arteriole. So efferent arteriole takes the blood away from the Bowman capsule, and finally the efferent arteriole opens into either the peritubular capillaries, which are found in the cortical nephrons, or either turn into vasa recta found in the juxtamedullary nephrons, and finally these structures takes away the blood into the renal vein. We also learned in the class that the renal vein is the blood vessel which carries minimum amount of urea in the human's body, and the hepatic vein is the blood vessel which carries maximum amount of urea. So just remember, if these things come in the MCQ, that the hepatic vein is the blood vessel which carries the maximum urea, and renal vein is the one which carries the minimum urea. We also learned in the last class regarding types of nephron in detail, that there are two types of nephron: cortical nephrons and juxtamedullary nephron. We learned that out of total total amount of nephrons, 85% is somehow the cortical nephrons and in the human kidney, and 15% constitutes the juxtamedullary nephrons. We learned that the cortical nephrons are smaller in size, and their tubules are much more coiled, and the loop of Henle is short, and the loop extends into the medulla for a short distance only. We learned that juxtamedullary nephron are larger in size, they show less coiling, and the loop of Henle is longer, and the loop of Henle dips deep into the medulla. We learned that the cortical nephron's major part is in the cortex region, and vasa recta is not present, but the peritubular capillary network is present, and the renal capsule is present in the outer cortex in the case of the cortical nephron. We learned that in the case of juxtamedullary nephrons, major part of the nephron is present at the junction of cortex and medulla, that is called as the juxtamedullary portion of the cortex, and instead of the peritubular capillary network, vasa recta is prominently present. And then we also learned the renal capsule in this case is present in the inner portion of cortex. We learned that the cortical nephrons are supposed to function when the normal water supply is there to the body, or we can say water level is normal in the body. Then then the cortical nephrons function. Whenever a human body faces the stress condition for water, then juxtamedullary nephrons become active. We learned that the cortical nephrons, in the cortical nephrons, diameter of the efferent arteriole and efferent arteriole is different. We learned that the efferent arteriole is wider and the efferent is narrower, but the sizes are similar in the case of juxtamedullary nephron. And another difference we learned was that DCT is located away from the mouth of Bowman capsule, as well as away from the efferent arterioles in the case of the cortical nephron, but in the case of juxtamedullary nephron, DCT is situated close to the mouth of Bowman capsule, as well as efferent arteriole. We also learned about in minor detail of the structure of the nephron. We learned that nephrons are also called as uriniferous tubule, and nephron is made up of two parts, glomerulus and the renal tubule. The tubule is 3 cm long and 20 to 60 micrometer in diameter. This renal tubule is made up of three parts: the proximal, the intermediate and distal part. The proximal part is made up of Bowman capsule and glomerulus. Together these two structures constitute the Malpighian capsule or the renal body, and then the PCT is there as the third part of proximal portion. The intermediate part is the loop of Hinley with the descending limb as well as the ascending limb. And we also learned that the distal part is the DCT, which is followed by the collecting duct. So this much we did in the last class, and we also understood the same thing from the NCERT diagram, that this is how the nephron looks like. The yellow portion we learned is the nephron. This cup-shaped structure is the Bowman capsule, followed by the PCT, then the descending arm of the loop of Hinley, ascending arm of the loop of Hinley, again there is PCT, and then we learned the wider tube here is the efferent arteriole, followed by the capillaries, and this narrower tube is the efferent arteriole. It may develop peritubular capillary network, or it may develop the vasa recta, and then the DCT finally opens into a large number of nephrons. DCT is open into the collecting duct. So this much we covered, and today let's start with the detailed structure of the nephron.